Hello, everybody. My name is Dr. Scott Egan. I'm an associate professor of drug discovery and medicinal chemistry in the Department of Chemistry and Biochemistry.
And I'm McLean Howland. I'm a computer science student, and I'll go ahead and get started with the presentation. So our research was working on identifying inhibitors of the SARS-CoV-2 virus using virtual screening techniques. So as you all know, it's a big pandemic. We've got 155 million infections, nearly 3.2 million fatalities, and here in the US, we account for nearly 18% of those deaths. And we've got more than 1,000 detected strains with new ones emerging all the time as the virus continues. So we were looking for ways that we could help with this. So the way we did that was by targeting a viral protease or a viral protein called MPRO. Now, a protease is a protein used in the replication cycle of the COVID virus, and it's a protease, so it's responsible for chopping up long strands of other viral proteins, like a big pair of molecular scissors. And if you take a look at the MPRO complex on the right, you can kind of see its quaternary structure. You've got, well, a big thing to point out there is the purple residues. Those are kind of like the tips of the big molecular scissors, and those go and cut the other viral proteins. And you can see in that figure, we've put a small weak inhibitor in there, the green molecule that is stuffed right in between those purple residues and will hopefully inhibit it. Another important point is that humans don't have any analog to MPRO. So if we find a molecule that inhibits it, hopefully it won't have major toxicity issues with humans. And we also started working on this research before a vaccine was released. And it's important to kind of have small molecule inhibitors or drugs to treat an infection of the virus, because people may not be able to get the virus or may not be able to get the vaccine, or they may already be infected with the virus. So it's important to kind of have both in your arsenal of treating this. The way these small molecule inhibitors work is by kind of fitting themselves into the active site of the protein right there, like with the green molecule in this diagram, and by stopping it from working. So it's kind of like stuffing a key into a lock and then breaking off the key, and where the lock is your enzyme or protein and the key is your small molecule inhibitor. So the way we went about finding a small molecule inhibitor for this protein was by using virtual screening techniques. So there's lots of databases on the internet containing millions of compounds. We chose one for this screen that contained about 100 million compounds that are commercially available, meaning that theoretically we could go and order them and have them show up to our door. So we took those 100 million compounds in the Zinc 15 database, and we applied some filters to get more drug-like compounds that look like existing drugs we have already, just so we can pare down that 100 million compounds into a more manageable 7 to 13 million compounds. And for this screen, I think we ended up with about 9 million compounds. So then we take those 9 million compounds and we run them through a computer simulation to estimate how well they bind to that active site of MPRO. And we get a big ranking, and then we take our top inhibitors and do some more tests with them. So now I'll talk a little bit about how we actually rank that binding energy. So we use something called a docking program to estimate how well the small molecule inhibitor or ligand fits into our target protein, which is MPRO. So it takes the small molecule and runs a bunch of simulations and tries to optimize how it fits in that active site of the protein, and spits out a number that represents the binding energy. And in this case, a more negative binding energy means a better fit because it releases more energy and is more stable. And we used a program called iDoc for this screen. Now, one problem that you run into with these virtual screens is that we've got 9 million compounds and we want to screen them all. If we were to screen them one by one, each one takes about 5 seconds. So 9 million times 5 seconds results in about 1.5 years of computational time. But obviously that's a little bit too long, so we had to find a faster way. The way we do that is by using distributed computing. So we've got big computer labs on campus. The one we use for this screen is called the MPAC lab. So we've got 30 high performance computers all connected on a network. And so I wrote a program that goes and distributes the 9 million compounds over those 30 computers and runs them all in parallel. So we've got one master computer that distributes the ligand files and tells the workers what to do. The workers run those docking simulations and then send the results back to the master. And once we're done running them, we compile the list of all 9 million compounds and sort them by the binding energy and take the top couple ones. I know I saw a couple computer science faculty and students in here. So if you're curious, I wrote it all in Python and just running on the MPAC labs using OpenMPI. Now the results of this virtual screen. So we took our big list of 9 million compounds ranked by binding energy, and we took the top 75 compounds. And then we subjected those to more filters. So we filtered out the top 75 compounds for things like toxicity, or things like functional groups that are almost guaranteeing toxicity in humans. And we also filtered out really similar compounds so that we weren't wasting effort testing, running more tests on really similar compounds, so we can kind of have a more diverse field. So that resulted in 28 compounds out of our 75. And you can see some of those examples on the right there. We've got the zinc database ID, you've got their energy score and their rank and structure. So once we had these 28 compounds, we went and tried to order them. So ordering them online, we got 20 of the 28 compounds on our door. So we packed those up and sent them off to Germany, the University of Lübeck, for more bioassay tests. And we only were able to get 20 of the 28 compounds because even though it's listed in a database that says it's commercially available, it's not always. You might not be able to reach the vendor. Sometimes they're just unavailable. So after we sent our compounds off to Germany, our collaborators over there ran a fluorescence resonance energy transfer based cleavage assay, which tests the inhibition of the small molecules we sent them against the cleavage activity of MPRO. And so in this case, 100% inhibition would be MPRO didn't work at all with our small molecule inhibitor, and 0% would be MPRO worked completely fine even with the inhibitor. So we identified two compounds, CP13 and CP25, that showed 45% and 35% inhibition of MPRO. You can see those on the upper right-hand corner with their structures. Then we took those two and subjected them to more intense computer simulations, molecular dynamic simulations using the AMBER-18 program to kind of further estimate how stable they would be when they bind to MPRO. And you can see that on the bottom right. You see the MPRO complex in white behind it and the inhibitor CP13 in orange right there. One thing to point out is if you see those purple residues that are the tips of the molecular scissors, it's kind of stuffing itself right near them and hopefully stopping them from working. So we identified two kind of top hit compounds, CP13 and CP25, but the 45% and 35% inhibition aren't necessarily potent enough to advance into clinical trials. However, these things do serve as good starting points for optimizing and creating more powerful compounds. One way you can do this is by attaching electrophilic warheads, just more functional groups to the molecule to make them more potent. And kind of a fun fact about this is that Pfizer actually recently started early clinical trials for a compound that targets the exact same spot that we were targeting in MPRO. And they found a compound that was potent enough to advance to the clinic. So it is pretty cool to see that this line of research could result in an actual drug. And you can see the structure of the Pfizer compound down below there. And notice they've added an electrophilic warhead, kind of like we were thinking about. So that it was a pretty cool experience. And this project was funded by the Bill and Linda Frost Fund at California Polytechnic State University and the European Commission through the SCORE program. Thank you. Any questions?
Thank you, McLean. That was great. So this paper is now open for discussion or comments.
Amazing. Will you work to increase potency or move on as Pfizer is on it? We'll probably move on as Pfizer is on it. They've got about 200 PhDs that came up with this and will continue to work on it. So I think we'll leave that to them.
see. Are there other questions? I have to see if I can, if anyone put their hand up. McLean, I have a question. This is great work. And you, for the MPRO, you mentioned that the compounds, the 2 compounds had 45 and 35% inhibition, and that wasn't enough. What is, like, how much inhibition is good enough? What would that be?
You know, I'm going to pass that off to Dr. Egan, who's the medicinal chemist.
Because it seemed good, Scott. Is that good enough, or is that not? It needs to be better.
It's not. It wasn't bad, but to get those numbers, they had to be at pretty high concentrations. So theoretically, you could have driven it much higher. You start getting into large amounts of drugs. So at the level that they were testing at, we'd really want something to be 80% or better, and that's where Pfizer's compounds are. They're up in the high 90s. So we gave it a good try, but we weren't successful. But anytime you do a project like this, you're always a part of a big team. Someone somewhere will find it. So we were just glad to contribute. But yeah, 80% is kind of the threshold there.
Any other questions or comments?
I just have to say out loud that I'm just so impressed at all of the I mean, everything that you did is incredible in such a short period of time, and just your presentation skills, everything is just very impressive work. So congratulations on that.
Yeah, thank you.
Great, thank you. So we'll conclude our first presentation. Yay. So we'll wait till 11:25 and we'll start our next talk. So a few minutes here, few minute break for our next talk.
Oh, I thought we were short on time, so I could have asked just another quick question.
No, we're still on. We have.
Yeah, I was like, I didn't want to know how much time we had. But what were they doing in Germany that we couldn't do here? Had you thought about that? What? Just talk a little bit more, if you can, about what their role was and what they did.
So I guess a big omission on our part is that our collaborators in Germany are the people who originally sequenced and figured out the structure of the MPRO protein, right? So they had copies of the protein and the ability to make it and do tests with it. So we sent off the compounds to them to do that. I think we were kind of lucky because they came out with that, they published it, and everyone went wild, and we managed to slot in and get them to help us out.
I see. That's great.
And there are also biosafety hazard concerns with having a virus like that on campus. So, and frankly, I don't like working with those viruses, so I'd much rather having someone with expertise on that. But you know, they were the ones that discovered it, so we didn't think we could do better than them. So we worked with them.
Makes sense. Thank you.